Hi, my name is Yulia, and I am postdoc at the Department of Imaging Neuroscience here at UCL. I will be guiding you today through one of the tutorials on OPM data analysis. And most of the function I showed you today is created by the fantastic OPM team here at UCL. And these tutorials is well created by them. So first of all, the goal of today is to guide you through the tutorial that gets from the raw data that is very noisy, looks something like this. It's more noisy if the subject is moving, to something presentable like this, where you can actually discuss what's going on in the brain. The data of the tutorial I will show you can find on this link, and the tutorial you can find on the second link. So to quickly describe the data in experiment, the single subject was measured with the latest Neuro1 Q-spin system that has 48 sensors. The subject was wearing individual scanner cuffs to hold sensor in the defined places. And the task was just an auditory rowing all ball paradigm. The more details you can find on the paper provided below, but the idea is a variation of the all ball paradigm. So what we will try to find today is M400 signal from the raw data. And importantly, to run this tutorial, SPM dev version is required, because I will show you a couple of functions today that are brand new and only available at SPM dev version. So let's start. I want to start with how to organize the OPM data. It's not yet agreed upon topic, so every lab does it a bit differently. Here we are trying to use a brain imaging data structure format that have a very specific structure to how you save the data. First of all, the data is usually named like presented on the slide. You have a subject with a subject number. You have some kind of description of the task. You have information about the which run is it, or sometimes either which session is it. Then you have a description of the data, so it can be raw data, for example, then the description part will be MEG, or it can be structural MRI data, then it will be MRI, and then you would have a type of the data it's saved in, right? So if it's a sensor position, it can be TSV file. If it's some kind of another metadata, it can be text file, and etc. And the function that structurized and shows SPM where all these files as a main data file and all the peripheral file are is called SPM bits file. And you fill in, which I will show you in a second, where all the data is, and it outputs the array of structures containing the directory information. So here how it looks. First of all, you will create a bit structure that contains all the information about the current data, which means the number of the subject, which subject the data belongs to, what is the task, because the subject can do a couple of tasks during experiment, and what number of the run it is for the task, and of course where the data is stored. And then the information about all the files, main file and all the meta files about the neuroimaging data will be created, will be put through the SPM bits file, and the directories of which file will be created. So let's go step by step. First of all, for the MEG data here, you can see I supply the category, which is MEG, and I submit the type of file, which is LVM, which is what Neuro1 system outputs nowadays. And the SPM bits file creates me the MEG directory. I also would have a position file, so the file that containing the name of the sensors and the position, X, Y, Z, plus the orientation of the center, of the sensors. And this file would have the format TSV. We will also have in our tutorial data the channel file, which is the text file that has the names of the channels, the type of this channels, that is magnetometers in our case, the units it saves the data in, and the status, whether it's a good or bad channel. And then we can also supply, and we will supply here in this tutorial, the MRI structure by the description of MRI. And it's all, and the SPM bits file function will output me the array of structure containing this directory information where all these files are saved, which I later can use to read in my data. So why is it such a complicated process? Because when you do the group analysis, you now can loop all of this and your future analysis. And the only thing in loop that needs to be changing is this bit structure, which is very easy. And you don't need to specify multiple times all the different file names for all the peripheral files of each subject, which makes the analysis neat and clean. So let's talk about OPM, SPM OPM create function. SPM OPM create function reads in the OPM data. It needs to be supplied the MEG file, the file with the raw data, and needs to be usually supplied the channel information, position information if you have it, and anatomical information as structural MRI scan, again, if you have it. We here will be working, as I showed before, with .lvm files. However, the SPM OPM create function also supports other structures, other formats of the file, like FI file that is provided by FILLines system, for example, or the old QSPIN function .bin file, or the one that gets from the circumagnetic is CMEG file. It's also can be you can also use it using the custom formats where you just apply the matrix of n channels and n samples. The example of which of this user case you can find on this link. So when SPM OPM reads in the file and it is provided the MRI information, it will automatically plot you and show you the positions of the sensors and the brain. So you can kind of quickly check whether everything is aligned. So what I'm talking about here is I'm talking about co-registration. It's how we know where the sensors relative to the head. So as I mentioned before, in this data, the subject was wearing the individual scanner cast that is done using the subject MRI. Therefore, all the sensor positions are defined. They're already defined in an MRI space. Therefore, I didn't need to do anything. I just applied my anatomical structural MRI data, and I supplied the position file data, and what was already all co-registered. And hence, we have this nice picture of the sensors relative to the head, and it makes sense. So if we don't have the individual 3D printed scanner cast, and in case of using the generic helmet, what is usually done is that you have somehow optical scans of the, so you need to use reference points from the helmet all the way back to the individual MRI to do the co-registration. So if you have some reference point from the helmet to the helmet on the subject, and then from the helmet on the subject to the subject head, and then from the subject fiducials to the MRI, well, then with all this transformation matrix, you can put all the sensor position into MRI space, and then SPM OPM create will give you a correct picture. So to do all this transformation matrix and to use reference points, we have the function SPM OPM opreg. And how to use it and the tutorial with the generic helmet can be found by this link, with this link. Again, this function needs 4 sets of reference points to have a transformation between all of these stages. That is how co-registration is done here at the UCL. But usually we would have individual scanner cast, and it's very nicely based on the individual MRI, so you don't need to worry about co-registration. So after we load in the data and we figure out where the sensors are, it's nice usually to check how the data looks like. So it's nice to see, first of all, the power spectrum of your data, and then to see the time course of your data. So for time course of your data, you can use SPM EEG review function. So you just apply here the D object loaded from the OPM, SPM OPM create. So you can see here that the data looks, the power spectrum looks very noisy, and this doesn't look like anything that screens or outputs. Sorry. So there is some specific challenges with the OPM data. And they usually comes from the 2 things. First of all, OPMs are magnetometers, so they measure everything. So with your brain signal, there will be usually interference noise. And if you're moving, there will be very high low frequency noise. So this is the spectrum from other data set, but you can see there will be some weird, there can be some weird peaks from the vibration. Here is a huge peak that you wouldn't see if you are not using optically tracked cameras to track where the subject was. Our favorite 50 Hertz line noise, and very, very much of the low frequency noise, which probably comes from the movements. And also, the other challenge with OPM is that usually you would have a low count, low channel counts, meaning that you can't use very effectively filters like signals SSS or Maxwell filters for SQUID data. So I will show you how to deal with all these challenges. But first of all, let's start preprocessing our data. First of all, we might want to crop our data to get rid of the breaks where participants were before the experiment start. So to crop the data, you can use SPM EEG crop function. The useful tip, I think, is that if you want some functionality that is not OPM specific, it's very useful to browse the SPM EEG library for the function, something like crop, or here downsample your data. So you might want to downsample your data. Here we downsample to 100 Hertz. And then the next step is usually identifying and annotating bad channels. And we now have a very nice functionality is our SPM OPM PSD function that usually was just plotting the power spectrum shown here or shown here. But now if you put the select bed equals 1, if you enable interactive mode, you can interactively select and annotate the bed channels. So here I will show you how it's done. So first of all, this is how you use the function. You provide all the necessary variables for doing power spectral density, calculating power spectral density, and we're figuring out, we are switching on the interactive mode. And here I can show you quickly. Sorry. Ooh. Yes, I can show you quickly how it's done. So here I'm evaluating the part with the power spectrum calculation, and you can see the spectrum we already seen. And some of the weird channels, the noisy channels, are having this, the data points are stars. So SPM, OPM, PSD function will identify and suggest which channels are bad. And they will be annotated in these stars. So when you have these suggestions, you can now drag and select the sensors that you think is bad. So here, for example, I magnify something to see the better the channels, and I'm dragging and selecting the bad channels. Here's the other one. This one that I suggested, I'm leaving because I don't think that's a big problem for this data. I'm leaving in. And you can see here, so these indexes will be all the channels that we just selected will be safe in this indexes variable. You can see it here. I will type in my command window indexes, and it will show me all the 9 channels we selected. So the next step that needs to be done and shouldn't be forgotten is to actually annotate the bad channels. So using these indexes, don't forget to find the name of these channels and select them as bad channels. Without this step, you would just have indexes of the channels you thought is bad without annotating them. So you need to use bad channels function to annotate the bad channels. And as a next step, after bad channels rejection, we usually bandpass or notch filter our data, depending on your task. For example, with the my audio task that I'm describing here, my filter will be from 2 to 40 hertz because I'm not interested in very low frequency or very high frequency, and I will also get rid of the 50 hertz noise. And then is the more elaborate filtering of the data. So as I showed you before, the OPM data is quite noisy because it's the magnetometers, and anything from the car passing by to someone closing the next door will be interfering with the brain data. However, this interference would usually have a very specific spatial pattern, different from the brain. An AIM filter, which is adaptive multiple model filter, designed by team and designed by team Tierney and shown in this paper, is using these different spatial patterns to distinguish between external interference and internal brain, and then get rid of external interference. So this filter is analogous to signal space separation filter or Maxwell filter used for squeezed MEG data. This method models brain signal and external interference as a linear combination of vector harmonics. And then you just regress out the external modeled interference. This is a very powerful filter. Again, all the examples and all the nuances are shown in this paper. What I want to say is it works best with 120 plus channels. And the channel here, I mean the axis of the OPMs. So we would have here only 64 sensors, but it will be triaxial sensors. So we would have 3 by 64 channels. The other thing I want to say is that if you only have radial sensors, this filter won't be as powerful as if you have at least one other axis, one other tangential axis. And the reason why is because the spatial patterns measured by the only radial sensor will correlate a bit with the interference, spatial patterns. And the other thing that this filter might do, and it can work as the TSSS filter works, so it also can include the temporal filter. And to switch on this temporal filter, the only thing needs to be done, and this variable corrlim has to be set to something like 0.95 or 0.98 value. So again, more information and why these are these numbers you can find in this paper. You can find in this paper. So if you don't have that many sensors, you can use SPM, OPM, HFS filter. So HFS is homogeneous field correction. Models only interference part of the external part of the magnetic field, and models it only either as a DC or the first gradient field. And when it models it using only the sensor orientations, it just linear regress out this modeled interference. This is how this function can be used, and this L parameter defines either the DC component is removed from the data, or DC component plus first order gradient field is removed from the data. After we applied, in our case, because we have enough channels, the IM filter like this, our data is clean. So here's how it looks after clean. You can see that there is not too many outliers. We got rid of this high low frequency noise. This is probably some alpha, nice peak on a 10 Hertz. And if you're satisfied with your power spectrum after your cleaning data, we can go to the next step. And the next step being epoching our data. For epoching our data, SPM OPM epoch trigger function is very useful. You only need to supply the trigger channels. You can also supply, if it's labeled condition, that it's tone. You can do baseline correction, and if you put 0 here, you don't do baseline correction. Specify the time window of your trial, and the function will epoch your data. After epoching your data, we can use SPM EEG average as a robust averaging tool that not only just blindly averaging it removes the outlying trials from this averaging. And let's see how it works. We can see here that we have a big evoked response near 100 milliseconds, which is called usually M100 response on the audio evoked field. So data was nicely clean. There is no bad channels. We can clearly see M400 or M100 response. Nice. So I want to show you a couple of more useful tools. First of all, if you want to analyze the topology of where the signal is, you might want to use SPM EEG convert to images function. What it does, it takes the anatomical sensor location sensor positions, and it creates something like this, which is now very useful for statistical analysis. So after creating these images, using anatomical location of the sensors, these images can be used in a group analysis, and anything that is available for MRI statistics can be used for these images. If you only just want to plot your nice topper plots, there is a couple of methods can be done, but here I show you the use of the field trip function. So first of all, you to use the field trip function from the D object that SPM use, we need to switch to the field trip structure, which is done by using .ftro method. Then taking out the anatomical sensor location and creating the correct layout, it can be all feed into the FT topoplot ER function that most of you probably familiar with. And here will be a very nice plot, but only useful for visualization, because here we extrapolated data to fill the whole circle. So it's a nice visualization, but for statistical analysis, it's better to use SPM MEG convert to images function. This is all that I wanted to show you. The source analysis tutorial will be held later today, I think at 4:30, where Megan will explain you how to get from the, for example, evoke data to the source level. And the only thing I wanted to show you as well is useful resources. So if you visit the GitHub of the Phil OPM team, there will be a lot of tools from different tutorials to some fancy registration methods and OPM analysis. And the papers which work I used is the interference suppression techniques and then adaptive multiple model by Tim Tierney. In this paper, spherical harmonics based noise rejection, you can read about the HFS filter, which is useful for low count channels system, and how to plot and correctly create a topographies. You can check the last paper I provide here. Thank you very much for your time. I hope it was useful, and I'm ready to answer any of your questions.